Hi everyone, Mrs. Belo here. In this video, I'm going to be going over the Cell Cycle Lab Part 1. So this is for any student that was absent when this was worked on in class. So what we're going to be doing is we're looking at onion root tip cells, which are, it's the root of the onion which grows into the ground. And as the roots are growing into the ground, that's where active cell division is occurring, and that's where we can see cells that are going through the cell cycle. Specifically, we're looking at this area of the onion root tip cell. It's a really thinly sliced portion of the root, and it's been stained and put under a microscope slide. And we're looking at this section here because this section is the root cap, which is just cells that are providing protection. But this area is where active cell growth is occurring. And we're going to look at those cells and we're going to count how many are in each phase of the cell cycle: interphase, prophase, metaphase, anaphase. And then we're going to put telophase and cytokinesis together because when you're looking at them under a microscope, you can't tell them apart. They happen at the same time. And once we collect those numbers, then we can calculate the percent of cells in each of those phases and how much time they're spending in each of those phases of the cell cycle. So the first thing is to identify all the cells that are, we're going to start with interphase, okay? And then we'll go down the line here and write down how many are in each phase. In this lab, I'm providing you accurate data and this picture, but when you do part 2 for the lab, you're going to have a picture that was taken under the microscope, and the data might be a little different. We're going to compare that to this data. So the first thing I'm going to do is count how many are in interphase. And in interphase, this is when the nucleus is still intact. This is when the cell isn't physically dividing in half yet, rather it's preparing to divide. So this cell would be a good example of interphase. The nucleus is solid, it's still intact. This one, we're going to count how many. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, I should have 20 unless I miscounted. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20. So that's 20 cells total. Now, when you're looking under a microscope, it's real life. These images aren't going to be totally clear. So just when you're looking at, for part 2, when you're looking at your image from the microscope, just count the ones that you can identify. Even looking at some of these, they don't all look the same, and that's okay. And sometimes that can make it a little hard to distinguish what's what. Interphase and prophase look very similar to each other. But the key difference is prophase, the nucleus is going to start to disappear, and you're also going to see the chromosomes start to become present. So this is a really good example of prophase. The dark little structures, that's the chromosomes. The DNA has condensed to form from chromatin into a chromosome, and they start to become visible, and the nucleus is also starting to disappear. So we have 1, 2, 3, 4, 5, 6. This one counts as well. 7, 8, 9, and 10. I know I was kind of going all over the place. When you work on part 2, I would count either I would go a column or a row at a time to make it easier. So we have 10 that are in prophase. Metaphase is pretty easy to identify because it is when all the chromosomes are lined up in the middle. So we have one here, so 1, 2, and 3. So that one's pretty easy because you can see the chromosomes are lined up in the middle and there's not that many of them. Next, we have anaphase, where they're being pulled away or apart from each other. We can see that here in this one and in this one. It's quite literally the next step after metaphase here, where they're being pulled apart from each other. And we only have 2 of those. And then the last phase we have is telophase/cytokinesis. And that's going to be this guy right here. You can start to see telophase, think 2. You could start to see 2 cells forming. And then eventually this will completely split and form 2 new cells. And we only have 1 of those. Okay, so we counted all the cells that are in each phase. And now we're going to do a little bit of math with that. So we're first going to calculate the percentage. Percentage is pretty straightforward. You're going to take the number of cells that are in that phase. So interphase, we had 20, divided by the total number of cells times 100. And the total number of cells, we have to add all of those up. So 20 plus 10 plus 3 plus 2 plus 1 is 36. So 36 is my total. So you can see in the equation here, total number of cells, those are all the cells added together. Number of cells in that phase, this is the number of cells in interphase. And then we multiply that by 100. So we do 20 divided by 36 first in the calculator, which is 0.555 repeating. We multiply that by 100, and I get 55.5. I'm just going to round that up to a whole number. You can use whole numbers for your answers. That would be 56%, and I'm also going to write that number here. Okay, so that's what you're going to do for the percentages. So do that for the rest of these, and then there is a key posted in the content library to check to make sure you got the correct numbers, the correct answers for these. For the second part of the calculations, you're going to calculate the amount of time spent in each phase. So that is the decimal percentage, so the percentage that we just got as a decimal, times 24. The reason we're using 24 is because that's actually how long it takes for one of these onion root tip cells to go through one round of cell division. It actually takes about 24 hours. And so we're going to take our percentage, which was 56%, and as a decimal, 56% as a decimal is 0.56, and we're going to multiply that by 24. And we're just going to, it's 13.44, and we're just going to leave that as 13 hours as a whole number. And then I'm going to write that here. So again, do that for the rest of the phases using this data here. And then again, check your numerical answers against those in the content library to make sure you did them correctly. And then these 4 questions here, you can use some of the stuff up here to help you answer those questions. These 2 you may have to look back in your notes to answer. And I do want to clarify, by not dividing, I mean the cell is preparing to divide. So you can look back in your notes in what phase is the cell, not physically, not in the dividing part yet, but rather when is it preparing to divide in all of those phases of the cell cycle. And then in what phase is the cell actually physically dividing in half. This you can read the background information at the top here to answer that question. I kind of already answered that at the beginning of this video as well. And then this answer, you can just look at the calculations up here. And then once you've answered all 4 questions, be sure to check these with your teacher to make sure that you have the correct answers so you can be better prepared for part 2 of this lab.